Hello, welcome to WSQ number 18, chapter 5, section 4. We're going to talk today about genes, DNA, and proteins. We're going to learn about what forms the genetic code, how a cell produces proteins, and how mutations can affect an organism. So let's jump right into it today. We need to start, first of all, just with a quick understanding of what are we talking about when we talk about the DNA code. Remember, if we look into the cellular level, we would find a nucleus, and inside that nucleus in the cell, that control center, we would find chromosomes. Okay, chromosomes are basically made of DNA, and each of the chromosomes in a human, we have 23 pairs of chromosomes. Each chromosome contains thousands of genes, and the sequence of those bases in a gene actually forms a code that tells the cell what proteins to produce. So at a basic level, the purpose of DNA is DNA tells our cells make these proteins. And those proteins ultimately determine what we look like, the size, the shape, the function, the features of the different parts of the body. And so genes are really important. Genes can be hundreds, if not thousands, of nucleotide bases or nitrogen bases long. Okay, even into the millions, and it could be as small as just a few hundred as well. So they vary widely. One thing we need to understand is that those chromosomes are made up of DNA molecules, but even at a smaller level, they're made up of, remember those things that we talked about, all DNA is made up of a sugar, a phosphate, and a nitrogen base, also known as a nucleotide. So when we look down at a gene, we're looking at a section of DNA that contains the information to code for one specific protein. And that, as I said, could be hundreds of nucleotides long. That could be millions of nucleotides long for a particular gene. There's also something else we need to know. There's a word known as a codon. A codon is actually 3 DNA bases. Okay, 3 DNA base codes that make an amino acid. So every 3 bases, let's say in this case TAC, on the genetic DNA information actually codes for a particular amino acid. Okay, for example, the amino acid, I think it's alanine or alanine, actually is coded from the codon CGT, so cytosine, guanine, and thymine. So anytime you see CGT in order, that is going to actually produce the amino acid alanine or alanine. And that's important because proteins are made up of those amino acids plugged together in order. So that's what the DNA code looks like. Okay, remember, please don't forget that there are 4 types. We have adenine, guanine, cytosine, and thymine. Those are the 4 nucleotides that make up DNA. And that's going to be important as we go forward. Now, quick question for you about DNA. I want you to read these following 2 sentences, and I want you to think to yourself, how are they similar and how are they different? Don't need to write them down. Just want you to see the similarities. The boy ran home with the ball. And here's the second one. Hob the Ram Yin Hot Well habit. So take a look at them for a second. So, what are the similarities? Well, if you look closely, you'll realize both of these have, I don't know if you may notice, 24 letters. In fact, if you looked closer, you would recognize that both of these have the same letters, okay, just in different order. Now, what are the differences? Well, this one makes sense. This one doesn't, okay? These have the same letters, the same number of letters, even to some degree. Look, we've got 3-letter words for the first 3. Okay, a lot of similarities, yet the order of letters greatly determines the sentence. So if you think of the sentence like a protein and each of these words like an amino acid, the place that the words get put or the place that the letters get put, the order and sequence does matter. So you need to understand when we talk about DNA, sequence matters, okay? So that question, does order matter? Absolutely. Sequence matters because sequence is going to give us ultimately the traits or qualities that we need to know. Now, if it was just DNA, that would be fine. We'd learn that and we'd be okay. And there's another nucleotide known as RNA. So DNA stands for deoxyribonucleic acid. DNA, remember, is double chain, double helix design. DNA codes for the proteins and is really the genetic information. RNA, on the other hand, is similar to DNA, except it has three major differences. And let's go through those. Number one, RNA only has one strand, one chain instead of two. Okay, so DNA looks like a ladder. RNA looks like a half a ladder. If you take a ladder and cut it straight down the middle, that's RNA. Okay, number two, RNA contains a different sugar molecule than DNA. DNA, in fact, deoxyribonucleic acid. That actually refers to the sugar. RNA, ribonucleic acid. So there's a different sugar when you look at the phosphates and the sugars attached with the nitrogen base. And thirdly, RNA actually has a change to its nucleotides. DNA's nitrogen bases are adenine, guanine, cytosine, and thymine. RNA, instead of thymine, we have uracil. So uracil instead of thymine. And uracil will connect to adenine. So just like thymine connects to adenine, uracil connects to adenine. So those three things are very different about RNA. Also, there's only one type of DNA, but there's actually two types of RNA. We have the first type known as messenger RNA. Messenger RNA is, its whole design and function is to literally copy coded messages from the DNA, so to go into the nucleus, make a copy of the DNA, and then carry that message to the ribosomes. And at the ribosomes, that's where that messenger RNA actually gives the coding for the particular proteins that are needed to be produced. Transfer RNA, on the other hand, carries amino acids to the ribosomes and actually adds the amino acids to the growing proteins. And as we'll see, it's like it brings the fabric or the cotton to make the shirt, as you would see. So oftentimes we'll see mRNA with a lowercase m, RNA, and you'll see tRNA like so. So you need to know both of those, mRNA, RNA, and tRNA. Now, with that being said, here is just a view of the two, and we can see the differences. DNA here on the left and RNA here on the right. And notice just a couple major differences. You can see the double two-sided structure of the DNA and the single structure of the RNA. The sugar doesn't show up here as well. It is still sugar and phosphate. DNA and RNA just have a different sugar. We can see that in the name. RNA is ribo. DNA is deoxyribonucleic acid. But the other big difference is, notice over here on DNA we have thymine, and on RNA we have uracil. So you need to know those three things about the difference in DNA and RNA. That brings us to protein synthesis, which is probably one of the most complex processes that happen inside of the cells of living organisms. And we're just going to barely piggyback. We're going to barely hit it today. We don't spend a lot of time on it. Basically, there's four major stages of protein synthesis, and I want to just quickly walk through them. And you could really break them down to just a few words. Step one, it starts in the nucleus. And protein synthesis, before we really get into it, this is the process by which the cell actually takes information from a gene on a chromosome and produces a specific protein in a ribosome. Okay, that's what's going to happen in this process. So we're going to start in the nucleus. RNA is going to come along. Messenger RNA is going to do some work, come over to the ribosomes. You're going to see tRNA working, and you'll see new proteins made. That's what's happening. Protein synthesis, the creation of proteins. So in step one, first thing we're going to do is you're going to have the messenger RNA production. So in the nucleus, the DNA, remember we said at one point the DNA would actually go through what's called transcription, where the DNA would split, right? And so as it splits, it does this during, if you guys remember, we did this at the beginning of interphase. It would split, and then we'd have free DNA to come on in and would connect, and you basically end up getting two brand new DNA chains, right, that are replicas of one another. That's why I call it transcription or replication. In the case of RNA, we have transcription where what happens, it just opens up and the RNA will actually come in and connect, as we can see here, to the side of the DNA, thereby making a messenger RNA. And so that messenger RNA ultimately is going to be the strand that will then travel into the cytoplasm to a cell and will line up inside of a ribosome. It'll attach to the ribosome, and then it becomes the genetic information for amino acids, okay, that are attached to transferrin. So here's the amino acid, and here is the transfer RNA. The transfer RNA actually bring the amino acids, and the transfer RNA connect by codons onto the RNA. Okay, so step 1, messenger RNA is made through transcription. Step 2, the messenger RNA actually goes into the ribosomes and starts the process of moving along the RNA strand in the ribosome. Step 3, let me get rid of this. Step 3, then what happens is, what you'll start to see happening is the proteins are starting to connect together. The amino acids are starting to connect together one by one. And as these will connect together, they're ultimately going to add to a growing protein that's going to get longer and longer and longer. And you'll notice, how do they know what amino acids to match? Well, they use these 3-letter codons, right? So as we look, the next set of 3 is GGC. So here comes the transfer RNA with that particular amino acid, the opposite, CCG, which will connect, and attached to it will be that amino acid. And so you're getting this chain of protein, which can be hundreds, if not thousands, if not millions of amino acids long. And ultimately, it'll continue to do this until it comes to a point where it'll have what's known as a 3-letter stop sign codon. So you actually have a codon that's whole job is to say, stop! And it'll stop the process, and the protein will end at that point. So that's protein synthesis in a nutshell. Okay, this process where you have messenger and transfer RNA and ribosomes working to produce proteins. Ultimately, then, these proteins will go out into the cell, and those proteins are able to be used by the cell for whatever particular function they were designed. There's a really good activity on phschool.com, CEP 3034, on protein synthesis. Would encourage you to do that. Last thing I want to talk to you about are mutations. Mutations can happen sometimes in a cell, and at genetic level, they can be where you have an organism's trait or phenotypes are actually changed or different from normal because of a change in the actual sequence of DNA. So let's say here's an original DNA sequence. There are 3 different things that can happen. Okay, you can have a substitution where one pair is actually substituted. Instead of the C and G as it was supposed to be, you get an AT, and that actually creates a mutation. You can have a deletion where you actually have a whole segment cut out. And remember, if that's gone, that's going to mean that this will be the next amino acid. It's going to change everything after the fact as well. Or you can have an addition where you've added an extra base pair. So whether it's a substitution or a deletion or addition, each of these are going to produce different codons. And these different codons, or 3 piece pairs, are going to produce different amino acids. Okay? And ultimately, the problem with mutations is they can be harmful because they may add non-functional structures, or they may actually do damage to the ability of that organism to actually survive. And we'll talk a lot more about mutations as we progress in this year. So that is it for WSQ 18. I pray that you got it all done, and you're going to do your summary and your questions now. Talk to you later. Bye.